neuroscience and beyond. Know more, get inspired. Welcome to the Neuroscience and Beyond podcast. My name is William Georgiev and I am a PhD student at the University Medical Center in Göttingen, Germany. Together with my colleagues Sabina, Christina and Lenny, we desire to bring science, and especially neuroscience, closer to the general public. In the following episodes, we are going to talk to various scientists about their inspiration to do science, their research project, and their opinion on current scientific problems. Today, our guest is Professor Alexander Gail. Professor Gail is the head of the Sensorimotor Group at the German Primate Center in Göttingen, Germany. His research focuses on the neural processes underlying goal-directed behavior. Professor Gail and his team investigate the basis of movement planning and decision-making in different areas in the primate brain. His work in fundamental neuroscience goes hand in hand with research towards modern neuroprosthesis and development of neurotechnology tools. Before we begin, I would like to mention that this podcast is separate from my research position at the University Medical Center Göttingen in Germany. This podcast is supported by the International Max Planck Research School for Neurosciences, as well as the Cluster of Excellence Multiscale Bioimaging in Göttingen. And now, please enjoy the talk with Professor Alexander Gail. Hello, Professor Gail. It's a pleasure to have you here today with us. And I remembered our first meeting. You perhaps don't remember, but it was during my interview for the graduate program here. And it was the first time that I met you, and now the roles are kind of reversed. I will have the questions and you have to answer, which is kind of a nice feeling to have.
Should I be scared?
I hope not. Thank you for accepting our invitation and being here. I would like to start with your past and your scientific path, so to say. You have a different story. So far, we had scientists that kind of started their career in the field of biology, but you studied actually physics, and then it's physics, and at some time you switched to neuroscience. Could you please tell us more about your ideas behind these decisions, and how did you end up actually doing science, neuroscience in particular?
Yeah, sure. Yeah. I understand that from an outside perspective, looking at a CV, that must look like a strong turn that I took. But for me personally, actually, it doesn't feel like that at all, because the interest in neuroscience was there earlier than my physics studies. They came during youth already, from popular science magazines and from an excellent teacher at school. So I actually strategically decided to study physics with the goal in mind to go into biomedical research. And that plan was even intensified during this time that in Germany back then we had this mandatory civilian service, where I worked with elderly people at their homes, and many of them were mentally affected and suffering from disease, neurological or psychiatric. And for me, it became very important and clear that I want to contribute to brain research. And I still decided on physics because structured neuroscience programs were not around at that time in Germany. And studying medicine, I felt, was way too much aiming towards being trained as a medical doctor for really working then as a doctor. And I wanted to become a basic researcher. And I had a better affinity to mathematics and physics than to chemistry, for example, because molecular biology was strongly in the rise in the 90s of last century, and I felt this would be also super interesting. But the intuition was more than towards physics. And I said, okay, I'll study physics, and then I'll look during my studies what opportunities are there to go into biomedical research. And that's what I actually did. During my main part of the studies, I already oriented myself towards biomedical research.
So you finished your physics degree in Germany, as long as I remember, and then you moved to the US. How was the research there in comparison to Germany, for example? And also, I'm sorry if I interrupt, but we had this nice comparison between the time points, because now maybe the research is not that different, but back then perhaps it was quite different between German and USA education, for example.
Okay, So you anticipate already my answer. That's exactly how I experience it too. So For me, it was just, a great opportunity. I felt totally thrilled to go to a place like California Institute of Technology, and I enjoyed it very much. I encountered there an atmosphere, a dedication to research, which I think is really unique, not in the sense that there would not be also other places, but for me it was new and it was exciting. It's not a university, right? So it's all about research, and no matter whether you go to the cafeteria or go to the beach for surfing or be somewhere else out in the evening, people are excited in a positive way about what they're doing and are interested in what you are doing. And that excitement for me was super motivating. People are energized about their goals. They think big often. They're not shy of thinking big. They're not so much focused on what could go wrong, but are optimistic that they will find ways of dealing with it. And this is something I really liked a lot. Now, I'm totally aware that CalTech is also not the average place in the US, and there's a lot of state universities that have to deal with much lower budget and things like that. But I do think that the overall atmosphere and the attitude towards science, that this is a widespread phenomenon. What I also liked a lot was how internationally diverse the place was. And this is something I was not used to from a German university. If I think of the labs where I did my master's thesis and my PhD, this was dominantly heavily by Germans and then maybe some international European students, but that mostly was it. And when I returned to Germany, which was always the plan after a few years, I felt this is something we need to change in Germany. And I have to say, without me being responsible for it, but I, of course, in my lab also made it sure, this has changed dramatically. When I arrived here after the US, all the emails were in German, all the documents, the paperwork was not prepared for international students. By now, at least here in Göttingen, this has changed dramatically. I mean, this is just became an international place, and I think it's a super good development. It's enjoyable to have so many people in the lab who come from a different cultural but also educational background, and I think that just adds to the whole adventure of doing science together, and I enjoy it a lot.
Indeed, I completely agree. I used to live in Berlin. I did there my undergrad, and then I moved to Göttingen, which is a significantly smaller town. But I was surprised from the diversity, cultural, international people who are studying here. Even in our program, we are 20 people in the graduate program from all over the world with different backgrounds. And this is one of the cities where you actually can survive without knowing German, which is also cool. Although I already know German, but
I still encourage students to take German lessons and a course to just have it easier for socializing even outside academia, because there is a world outside academia. Absolutely. But yes, it's easy to survive here without.
Absolutely. Yeah. But what made you change your mind and go back to Germany instead of staying in the US, for example? And in particular, why did you choose Göttingen?
Yeah, so again, it was not really a change of mind. It was planned that I would return after a few years. It sounds like that I have planned everything in my life. That's for sure not the case. But it's hard to predict how things work out. But it worked out for us. So when and I say we now because I went there with my partner to the US, we both find something super interesting for us. But it was clear to us that we want to return after a few years and start a family with having kids. So we are married by now and we have kids, but we wanted to raise them here in Germany, being closer to the rest of the family. That was one of the main reasons. Research-wise, I would easily have enjoyed also working in the US, but family-wise, I preferred it here and I don't regret it. It was important to be close to the family, and it still is. I mean, my parents have passed away by now, but that was one of the reasons why we returned. Göttingen, that was just the right offer at the right time, right? Sometimes these things happen. If at all, maybe it was not the right time in the sense that it came relatively early. So I was there less than 2 years in my postdoc, and as in monkey system neuroscience, this is nothing. So I got the offer when I was still in the middle of it. For those in the field, they would know what I'm talking about. I only had finished my first animal, and you need a second one, and that is a very unpredictable timeline. So I got in contact with the people here from the Bernstein Center, Theo Geisel back then, who was still director at Max Planck, and Stefan Treue as the director of the DPZ. They basically came to my poster at a conference, at the Computational Neuroscience conference, CoSign, and I was like, why the hell are they interested in my poster? I'm not working in their field, and it turned out they were basically headhunting. And then they pointed to me that opportunity for an independent group leader position. And at the DPZ, which when I visited then, I was super excited about how professional the whole setting is when it comes to animal research, right? All the competence that is there from the caretakers, from the veterinarians, from all the personnel organizing that. So that for me was a very big argument, but also, of course, the Göttingen Research Campus. I mean, this is just amazing how much neuroscience there is in a not so super big city, as you just said yourself. Of course, it's a big and traditional university and a very good one, but still, neuroscience has, even on top of that, some extra value here and a very nice community. And this combination made me consider keeping my postdoc time shorter than I originally had planned. I negotiated to push the starting date as much as I could, but of course there were limits. And I managed, fortunately, to finish at least my physiology project to the point that I collected all the data, and then you can carry your data with you and then finish the stuff afterwards. But yeah, that was a very exciting opportunity, and I have to say I never regretted it. This has worked out perfectly well for me. It was not tenure track at that point. I felt a significant risk in that because monkey neurophysiology labs are quite involved. We have these, lead times of several years before you have your animals trained, your task setups, your programs developed, and whatnot. So a non-tenure track position of 5 years, I felt is a risk, and it made me nervous, I have to admit. There was no doubt. But I also, at the same time, experienced the people who invited me to be very responsible people, and that I got confirmed. They really were aware of that and worked towards solving it, and they managed to solve it, so that in the end it was the right decision, and I'm super happy to be here.
Once again, we hear about the right timing and the right opportunity at the same time, which is maybe a nice recipe for success, I must say. Beside this insecurity of the tenure track, how did you felt in terms of your scientific experience to lead a group back then? Perhaps it was also challenging and new to you.
Absolutely. This was what we in Germany call the Sprung ins kalte Wasser, right? You were just confronted with that situation from one day to the next. I do think that, let's say, in the traditional German system, where postdocs often are for several years in a lab and have much more responsibility also towards running the lab, the group, helping with teaching, supervising of students, all that was hardly the case at CalTech. At CalTech, in this lab where I was, when you entered as a postdoc, you were responsible for your project, you worked towards this. There were hardly any PhD students around. It was almost exclusively postdocs in this lab. And so you were just responsible for your own thing. You learned a lot about, doing surgeries and whatnot, the things that I hardly had opportunity during my PhD. And it was sink or swim, right? You had to learn it, otherwise you could not have continued with your work. But there was little opportunity to supervise bachelor level, master level, PhD level students. So that is a component that I actually had more during my PhD in Germany, the physics department, because teaching there traditionally plays a strong role. So I was tutoring courses. I was taking care of practical courses. I was supervising students in the lab. So actually, I profited more from my PhD time than from my postdoc time for this, which is certainly also helpful to have additional experience as a postdoc. No, this was then a hard cut because all of a sudden I had to hire the people. I had to decide on whom I want to work with. I had to decide on the projects. Yeah, it's something that you don't really know well in advance how easy it would be for you. I think I was blessed with 2 students who started with me who were super motivated and were excited to start something from scratch, right? This is, not everybody is into, starting something with an empty room and say, okay, this is our lab. Doesn't look like a lab, but we'll make it look like a lab. So, and I had people who were into it, right? And that worked nicely. There's other people who are really super good students, but they preferred to come into an established setting and with a more narrowly framed goal. And that would have been difficult at that time. So again, it's a little bit of luck there to run into the right people. And then soon, of course, we hired additional ones who could then, start from what we set up already. And there we were much more diverse. Also, the third student already was international from the Far East. So it quickly started that we had a much bigger diversity in the lab. And it was good that we had established a setting that would function well and people could just use it. So that helped a lot for new students to join then.
Yeah. Challenging but successful, as we'll discuss later, perhaps.
Before, I have to add, I had experience with setting up a lab actually also from my PhD time, which is also not the typical case. Because at the time when I started my PhD, we basically completely rebuilt the electrophysiology setup in that institute. So during my PhD, I started also from an empty room and built a multi-channel recording system for awake monkey neurophysiology. There was experience there before, but we started from scratch. So I went through all these steps of programming, graphics boards, very low-level machine, close-to-machine code. And I partly redid this again during my postdoc, and that was actually the part that wasn't very easy when I started my own lab. I really knew what to do. Every component in the system I knew from the bottom of my heart and could work with it. And that helped me also to train the students. If I hadn't made this experience before, I probably would have struggled quite a bit with, dealing with all these details, how to do real-time programming and stuff like that, which is needed in our case.
So I was about to ask you exactly this physics and then neuroscience, but you already mentioned. So during your PhD, you kind of built up this electrophysiological setup. But what was the physics part of it, the biophysics and the coding, the hardware, more or less? Or?
Yeah. So. Basically, if I may rephrase the question, is did it help me in the end? Did my plan work out that using physics training? Now, true physics you're not really doing in this context, although we always made sure in Marburg in the physics department to sell it as physics so that we can hand in the master thesis or diploma it was called back then in the physics department and so forth. But strictly speaking, of course, we were not dealing with the problems of physics at that time, right? Semiconductor physics and whatnot. That was totally not relevant here. But of course, you get this highly formal training of, on the one hand, being trained like a mathematician, but being much more practical about it than a mathematician. So, this applied mathematics, this formal thinking, this logic thinking, this rigor with which you apply the logic of mathematics to problems, I think helps in dealing with complex systems. And I mean, that is a inherent part of physics, dealing with complex nonlinear systems, and the brain is such a system. So in that sense, the way you conceptualize things, the way you describe signals that you're recording and how they could be, understood from the perspective of a complex dynamical systems, of course, that all helps you. On the more practical side, because you jumped in when I talked about setting up the lab, I have to say in today's physics studies, it's not automatically that you would learn about electronics and all these practically relevant things. Also, coding back then was not part of the physics studies, right? I took the courses back then based on Linux in programming. Basically, it was autodidactically that you then learn programming really hands-on with the task that you try to solve. I took in the evening courses on electronics because they're not part of the curriculum. But there was this one guy at the physics department who offered in the evening those courses in electronics. So I went there with one of my friends and we attended those courses as well. So these are the things actually that came on the side. Physics is really about,
more the thinking
modern,
behind it, the logics, the modeling.
Yeah, exactly. So in that sense, that only indirectly helped. I should not say only, because this is, of course, very important. And then the practical things that I just autodidactically learned on the side.
Great.
But you have I do think you learn as a physicist a real fundamental way of how to address complex systems and also complex problems, and how to try to reduce them to solvable sub-problems and things like that. That's very inherent to the training in physics.
Yeah, that's true. I believe physics, mathematics, this kind of thinking, it helps in general in science, not only in computational science, especially now when we see all of this machine learning, artificial intelligence kicking in, modeling here and there. It's definitely a must to have this knowledge, or at least to more or less know about it, to be aware of these concepts, right?
It's extremely helpful, I would say, yeah.
Yes.
Yeah.
And now you are already well-established group leader at the German Primate Center, dealing with the sensory motoric of the brain and the central and peripheral nervous system. Could you please elaborate what is your research about?
So in very general terms, our research is about how we use sensory information from different sensory modalities, what we're seeing, what we're feeling about the status of our body, and how we translate that into a plan for a movement. And when I say plan, I mean actually the thing that happens prior to actually overt movement happening. So when we're still maybe deliberating about whether we do a movement to the left or to the right, in that sense, planning movements also entails deciding among different movement alternatives. And at a certain level of abstraction, that would also be decision-making in a classical sense, because the choice between left and right in our experiments, we sometimes make more or less attractive to the animal by changing the value of reward that is assigned to the different targets. But we really try to understand how this integration of multisensory information, which primarily happens in the posterior parietal cortex, where the different streams of information come together, it's called multisensory integration cortex, how that information is handed on to more frontal lobe structures of the brain, particularly the premotor cortex there receives a lot of input from parietal cortex. And then from there it goes towards motor cortex, so the part of the brain that really projects them down to the spinal cord. And as you might be aware, any movement of our periphery can only happen, the muscle contraction can only happen if motor neurons in the spinal cord become active. Mm-hmm. So the real output stage of the brain is the spinal cord motor neurons, but our interest is the stage before that. At the level of cerebral cortex, what are the neurons telling us about a movement that is immediately pending, that is coming up, what the animal has decided already to do or is about to decide what to do, and how do different parts of the brain contribute to that decision process. That's our core interest. And then, of course, that comes with a lot of subvariants, but that's what we want to understand.
We have mentioned already several times animals, and that we're also at the German Primate Center. So you're studying these problems in monkeys. do you also use patients or human subjects? And also maybe for people who are not aware, why do we use monkeys to study things that we want to understand about humans, for example?
Yeah, absolutely. So we do also study some of our research questions in humans. This is typically behavioral studies. So we're asking them to perform certain types of movement tasks or decision-making tasks. These days, even in interaction with others. So that's one of the interests that we're also having. But the type of information that we want to understand about the brain is something that depends on the activity patterns that we can observe in single brain cells, in single neurons. And to record their activity, we need certain types of probes, which you need to insert into the tissue, otherwise you would not be able to collect these signals precisely. And from those techniques that are available, I mean, there's fortunately a lot of techniques that can be applied in healthy humans without being invasive, without causing any damage, to register brain activity. But from those techniques available, none of them actually has a chance of seeing these very local, precise signals. So what we're seeing when we stick an electrode on the scalp is the so-called EEG, electroencephalographic signal, that is basically also an electric signal. So in that sense, it's as fast as neural activity. So we don't have a problem with temporal resolution in principle. But of course, it's a signal that is summed up by millions and millions of neurons. By the time you see it there on these electrodes, you're basically integrating a lot of activity already from large parts of the brain. And in that sense, the changes that you're observing there are also on a certain temporal dynamic that is not so super fast, but okay, reasonably fast. There's at least no time lag. But spatially, it's just way much more imprecise than anything you can record from inside the brain tissue. Other techniques like functional magnetic resonance imaging, right? They can resolve the 3D structure of the brain. They're not just on the surface, but they're very indirect measures in the sense that they're not recording electrical activity. They're measuring the change in the oxygen level that you're having in the blood. So this is something that changes with the activity of the neurons, so that they take up energy and use energy, and you can measure this in how much oxygen is bound to the molecules in the blood, and these machines pick this up. That's a very nice technique because you can scan the whole brain at the same time. You can do it in healthy subjects. You're not suffering any damage from it. But of course, it's indirect. It's slow because metabolic changes take a few seconds before they really become effective. So in that sense, you need to play a lot of tricks to get a reasonable temporal resolution, but it's also always limited. And if we look at things like the motor production that I'm currently doing, the speaking to you, right? The number of movements that I'm doing per 100 milliseconds or so, it's just immense, and you would never be able to really see that easily with these types of techniques. So if we at the moment that we agree that we want to understand certain brain functionality, and that we want to understand it at a level that is precise enough for really getting down to the circuit level description of things. And with circuit, I mean the neural circuit, the neural networks that basically are responsible for that function, then I don't see a way around of recording also from the individual neurons at that level. One can do this in human, but of course, since it requires opening of the skull to insert the probes, it's highly invasive, and therefore it's only done in the context of diagnostic surgeries that some severely affected patients undergo. For example, epileptic patients who are not responding well to medication. Sometimes they undergo diagnostic surgeries to find the focus of their epilepsy in the hope that by removing that part or silencing that part, that they might suffer less symptoms. And opportunistically, sometimes researchers can jump on these approaches and insert more experimentally oriented probes, but they're also, of course, connected with the hope that at some point they will become relevant for diagnostic purposes. But this opportunistic way of collecting data is possible in humans. There's another example where paralyzed people by now are, if they volunteer to contribute to research, are in the US at least, not in Europe so far, allowed to undergo surgeries for chronic implants with microelectrodes to do these types of recordings. But that's at the moment happening exclusively in the context of brain-machine interfaces and neuroprosthetics. Mm-hmm. But these possibilities are out there and we're open to those, but the very well-controlled experimental approaches that we need for doing the type of research question where you have an experimental design, where you can manipulate and at the same time study a healthy primate brain, that unfortunately is still only available in animal research. Now, of course, there's other animals than monkeys that are also used in neuroscience, and that's a great thing to have, because I think in the end we will need a spectrum of methods and approaches to really get a grasp of what central nervous systems are all about and what the brain is all about. But of course, the primate brain comes with certain peculiar things that are unique to primates, and that starts out from their visual system, the way their retina, so the light-sensitive sheet in the back of our eyes, is composed. It's continues with the huge early visual system that we're having at the cortical level, the huge expansion that we saw over the course of evolution in primates when it comes to the so-called association cortices. So these are, for example, the parietal cortex that I mentioned before is an association cortex, but very particular to primates, of course, prefrontal cortex. You don't find that in rodents anywhere close to what you're seeing in primates. And it serves an important function and certain cognitive capabilities that when we try to see them in non-primate species, they are very difficult to find. And sometimes when they are there, let's say in birds, which have often surprisingly high cognitive skills, then they are building on a completely different neuroarchitecture of the brain. So the bird brain is very, very different from the primate brain. And some of these cognitive skills that they're showing apparently came into existence in a very different way. Mm-hmm. So if you're doing it for the purpose of not only finding one specific neural network that would be able to have the properties that you see in cognitive behavior, but you also want to have the potential of using that in a translational way for improving the treatment of neurological disorders, the treatment of mental disorders, then it narrows down to this one model system, and that is the primate brain. Now, of course, there's many different primates, and then we can talk about why rhesus monkeys or marmoset monkeys or not other monkeys. Yes, but that's already a detail, I would say. Mm-hmm. The main message is the combination of understanding the brain with a decent complexity and with the potential to transferring that knowledge to humans. For me, that narrows down to the monkey model, and that's why I started working with monkeys. Mm-hmm. I started out with caretaking for elderly people and saw how big the suffering was in their families from mental problems that they had. And I was motivated to do something about it. And that's why I ended up with monkey research. Not because I woke up one day and said, oh, I want to do monkey research. No, it is a thought process and ruling out of other possibilities, and then you end up exactly there.
It sounds very plausible and, as you said, logical. Before we continue with the next question, I just want to clarify. You mentioned this electroencephalogram and functional MRI, which are non-invasive, and some of other methods are, as you mentioned, invasive. And maybe another one is the deep brain stimulation that Parkinson patients usually get as a treatment in a very late stage if there is nothing else possible. But could you please describe how does your setup look like? So you said chips and monkeys, and what do you do with this? How do you implant the chips in the certain brain region? Do you record where the monkeys are doing some tasks or they are freely behaving? How exactly is this working?
Yeah, I'm happy to elaborate on this a little bit. Actually, from the things you mentioned, basically we're doing everything. So I would say we're doing still what I would call the traditional approach, and that is that you train your animal to conduct a certain task, typically on a computer screen. So basically they're being shown stimuli on the screen, and then they have to react in a specific way. Let's say one of the workhorses of our field is the so-called center outreach task. So the animal would just put its fingers on the center of a touch screen, and then we flash a target stimulus somewhere in the periphery. Mostly the animal is required to keep the direction of gaze in the center of the screen. We call that ocular fixation. That's important because some of the structures that we're recording from, they depend on the direction of gaze and how they encode information. And then by means of this instruction, you tell the animal, please make a movement to the left or to the right or to the top or to the bottom. Now, in our case, as I mentioned, we're interested in planning activity, so we don't typically allow the animal to right away move whenever a target pops up, but we basically instruct the animal, hey, your target will be on the left. Mm-hmm. And only upon the second instruction, which might happen in 1 or 2 or 3 seconds, then you're supposed to go. And the animal learns that. They do a so-called memory-guided center outreach task then. So they get it flashed, wait for 1 or 2 seconds, then they hear a beep or something, and then they go there. That helps us to separate the visual processing of the instruction stimulus and the processing that happens in the context of the movement itself. Mm-hmm. And in between have a phase in which animals know already what they want to do, but the brain activity is not at the same time processing the visual input or producing the motor output, but it's in this, let's say, mm-hmm. cognitive state of idling, having a plan already what to do, and this is what we're often analyzing. Now, for being able to do that, of course, the animal must have learned to do that task in front of a screen, and we need to have a way of getting access to their nerve cells. And in the classical approach, we do this with a so-called craniotomy. So before that experiment, in a sterile surgery, so we do an opening of the skull and put an artificial closure on it. So we call it a chamber because it has a little bit of the shape of a cylinder that makes it easier for applying then later the measuring probes and also for keeping the inner part of it clean. But this is an implant that you apply during a sterile surgery, but then it stays on the animal for many, many years. And you keep it closed when the animal is together with its peers in the group. We group house our animals, so there's other animals around, of course, who can touch that and inspect that maybe, but they actually don't care too much about it. After a few days, they don't care at all anymore. But with this, we have then the possibility, once we bring the animal from its group housing facility into the lab, that we can just open that plug, basically. And what we're looking then at is no more bone, because we removed that, but we're looking at the skin that protects the brain. It's basically like an envelope around the brain. It's called dura mater. And that is a skin that is penetratable to super thin probes. So we have microelectrodes, we call them. They are basically very thin wires insulated with different types of materials. In our case, mostly it's quartz glass. Have a sharp tip, and you can insert them basically like a very thin needle. The outer diameter of these electrodes is like a human hair, so they're also fragile. So you basically protect them with what we call a guide tube, a little stainless steel metal tube that's just hovering above the dura. And then the electrodes are computer controlled and advance through the dura, through the skin, and then enter basically cortical tissue. And for this, the animal, of course, needs to hold still. So this is the traditional approach. The animal's head is stabilized via the implants that are on the head. And the animals get accustomed to this over weeks and months. That's long training. So typically, I would say at least 1 year from an animal that is naive about the purpose of the experiment to an animal that knows how to do this task in front of a computer, get accustomed to sitting in these so-called primate chairs, getting accustomed to the probes being applied, although they can't feel that. The brain itself does not have any sensors, so you don't feel if you insert anything in that tissue. But also the thing that we are inserting is so thin that you would barely feel it even if there was sensors. And that's, of course, on purpose, because we want to record from the intact brain, right? We don't want to damage this neural circuitry, so the probes need to be very fine. So that's kind of the classical approach. And then you mentioned already also that there's other ways that we are working towards, and that is by now we are also having animals where we don't insert those probes on a daily basis and remove them at the end of the day and close the lid, but where during the sterile surgery we insert so-called arrays of microelectrodes. Basically, this is a bundle that is arranged in a specific way, relatively small, let's say 4 by 4 millimeters, and then these small individual microelectrodes stick out of this ground plate, and that can be then inserted during the sterile surgery already. So there we would not only do a craniotomy, opening the skull, but we would also do a so-called durotomy, so opening the skin that protects the brain to push in these microelectrodes. And they come out with very thin and soft wires then to connectors, and we can seal everything hermetically. And by the time that the animal wakes up and is released from the surgery, basically everything is covered except for a little connector. And then we can basically in the lab plug in the connector and get the electrical signals from those implanted chips. Now, the big advantage of this is, of course, that you can achieve many more electrodes in the same time. So we record not just from 1 or 2 electrodes that you inserted on a daily basis, but you record from hundreds of electrodes if you want. And that is, for example, necessary if you want to use analysis methods that in real time extract larger patterns of activity, for example, to control a neuroprosthetic device, to let the monkey use its brain activity to control its environment, for example, a prosthesis or an artificial prosthesis on the computer screen, then you need activity from many more than just one single neuron. It just gives you a complex enough pattern to have enough information contained in that. And so that's one of the reasons why we do these chronic implants. And the second reason is, and that's something I'm super excited about recently, is that we can combine it with wireless recording techniques. So we can replace the tethered recording, the wires that we hook up to those connectors, but put instead either a system for data logging onto the animal. That means we have a little bit of an electronic board that writes the signals onto an SD card, onto some memory card. And then at the end of a recording session, let's say of 1 hour, we take out this card and feed it into our computers and can basically analyze what we've recorded in the last hour. Or we also have true wireless systems where there's a real-time transmission of the signal from the monkey's head to our recording computers. And for this, you also, of course, need chronically implanted microelectrode arrays, because that now allows us, and that's why I'm so excited about it, to record from these animals while they're not sitting in front of a computer, but while they're walking around in a larger enclosure. And we built such enclosures, and we have now data from animals walking around, making decisions in what I consider ecologically much more relevant settings. They're pulling down grapes from an artificial branch that we are hanging in those setups. They are, finding food in little so-called kongs. These are like plastic things where they have a hole and they poke in there. They're foraging on the floor because we spread wood chips and we're hiding some treats in there, so they do what they would do on a meadow also when you observe them. And that's why I'm so excited about it, because we're observing now behaviors that were never accessible to studies in the lab with high bandwidth neurophysiology. So with this approach, I think we're now getting into a new era of system neuroscience, where a totally new set of behaviors becomes accessible to our research question. And this is the type of decision making that in the end I want to understand. not necessarily how an animal picks between a red and a green dot on a screen, but how an animal decides to forage for food in this corner of the room versus that corner of the room. So that's why we have these different recording techniques for the different types of research questions. But yeah, that's what it takes to get there.
This is fascinating that we are at that age where we can explore the real behavior, maybe at some point even in the more natural context outside, but perhaps this is for the future generations, I don't know, or maybe we can experience that.
So when it comes to the behavioral part, I think that's happening
this
already.
Is happening, yes.
So we do actually the type of food dispensers that we're building in the lab that are computer controlled and that would release food upon a certain behavior of the animal. These are feeders that we also develop for field sites. So the German Primate Center, as you might be aware, has field sites in Senegal for baboons, in Madagascar for lemurs. And we put testing boxes there for cognitive tests. And when I say we, I mean my colleagues from behavioral biology, but we're collaborating with this. And this is part of this idea of moving to much more ecologically relevant behaviors in the lab. I think when we want to understand primate brains, we need to have in mind what they have evolved for. And the most direct answer to this is, well, observe what animals are doing in the wild, in their life, when they're acting in the context of their social network with the constraints that nature puts on them. And we in the lab over the last 40, 50 years made huge progress in understanding what are the constraints of, let's say, visual processing or solving certain types of abstract decision tasks. And I think it's a very exciting time to bring this knowledge together. And that's something that with the recent collaborative efforts that I started is exactly what we want to do. Let's understand what we've learned from the lab work about constraints and perceptual capacities and how that translates into what animals can or cannot do in the real setting in the wild when they're together with their peers and need to survive. And in the opposite direction, what types of behaviors do we observe there in the wild that we then want to understand and be able to study also with means of neurophysiology. And that's why the setup that I mentioned in which animals can walk around is actually a setup where we can also have more than one animal in there. And we did the first behavioral recordings where two animals are foraging together on the floor. And then we see how the activity of the one animal affects the activity of the other. So we see these mutual dependencies and the dynamics of the behaviors. And I'm super excited to now also get neural data from this behavior.
So something to mention, I think as long as I know your projects, you record from the brain, but you also have cameras in this setup. So you can see also what the animals are doing exactly, and are they alone, are they with their peers? But what I wanted to ask, when you have these chips in the brain, how many cells can you record simultaneously or circuits? And at the end, this is a huge amount of data when you have animals behaving the whole day. How can you analyze this data? And what can you disentangle from the behavior that they're reaching something, that they're doing some other task? How do you differ and say, okay, here we have this, maybe you combine it with the camera and look at the recordings at that time point, or how does it work?
Yeah, that's a very good question. And you probably know whenever a scientist says this is a very good question, it means I don't have a good answer yet. So we're asking this ourselves. That's why it's a good question. No, of course we have ideas about this. And in system neuroscience, I think we have a tradition of recording not just the neurophysiological signals, but also the behavior in a very precise, timely precise, but also otherwise precise way. Let's say vision research, right? It was always a standard from early on that we track at least the eye movements or teach the animal to withhold eye movements for a certain amount of time. And if you want to train an animal to withhold eye movements or do certain goal-directed quick eye movements, so-called saccades, if you want to train that with means of reinforcement learning, so rewarding for the correct behavior, you need to have a way of registering this behavior with super high precision, because otherwise we are not aware of our eye movements typically. So unless the consequence to an eye movement happens immediately, you can't learn the link between what you're doing and what happens and what you're supposed to do. So it has a big tradition in system neuroscience, and this was the first thing I also learned when I studied vision, because I started out in visual cortex with my career. Now that tradition we want to continue and also transport into this newer approach where we have much freer behaviors. So where the idea is not to train the animals to show certain types of behaviors on request and precisely in time, but basically let the animals do what they naturally would do. So cut down dramatically on the systematic training that is imposed on them. Of course, they still need to learn something, how to go into this room and get out of it again. Going in actually was not a problem. Convincing them to come out again was a bigger challenge. But now we want to let them show whatever behavior they would like to show to solve their task. But of course, we still want to know down to the level of millisecond, 10 millisecond precision of what their behavior is to link it then to the brain activity. Otherwise, we're completely lost. Mm-hmm. So we need to register their behavior with this precision. Mm-hmm. And you mentioned already, we have a lot of cameras in place. In our case, at the moment, we're working with 18. Other labs started out with more than 50 cameras. I think, fortunately, we don't need that many. We can get a 3D reconstruction of the movements of the monkey with much fewer cameras. We record at the moment with 100 images per second. So we have every 10 millisecond a readout of the full body pose and can do the 3D reconstruction. This is a painful amount of data on our hard drives. So we're talking quickly several tens of terabytes, and our IT department starts complaining. But the good thing is that in our setting, actually, we can compress those video massively. So I'm not so concerned. And we might be able to give up on some of the time resolution at some point. But at the moment, we're collecting it like this. And then now comes, this is all basically technology, and we found ways of solving it. I'm not so concerned about this. But the conceptual thing that you mentioned, how can we ever make sense, given that the brain activity as such is already complex? Now the behavior also becomes complex, and will we be ever at the point where we can make draw meaningful conclusions out of these two pieces of information? You asked how many channels we're recording, how many brain areas. I think for this type of approach, actually, it's good to have many. So it's not it is, of course, technically a certain burden, and there's limits to it. But for making sense in the end of the signals, I do think we need the larger scale signals. And we are at the moment recording from posterior parietal cortex, from primary motor cortex, from premotor cortex, and from prefrontal cortex in one animal that we just implanted a few weeks ago. This is like 512 microelectrodes. Mm-hmm. And we register them with full bandwidth, so 20,000 or 30,000 samples per second. So we have all the resolution to then extract single neuron spiking, but we can also use somewhat slower signals like local field potentials to analyze and extract from these data streams. That all is just numbers. The real question will be, and I think this is an unsolved question, how good will be our mathematical models and approaches to extract what is now really the driving force behind a certain type of behavior. If you start from the neural activity, now which neural activity drives what type of behavior, or the other way around, which neural patterns are associated with certain types of behaviors, no matter whether they are driving or just a result of that. Because the big challenge that we already have in sensorimotor research is we are poking into a closed loop system. So during continuous behavior, the brain is producing output and receiving input, and part of the input is the consequence of the output that I produced. But on top of this, there is other input because the environment might have changed. So I experience changes in my sensory signals because I'm moving. So whatever the sensors from the muscle send back to my body will change, of course. Now, theoretically, the body can predict that. We have so-called internal models that might have learned what the consequence should be, and then often we're only sensitive for deviations from that prediction. But of course, the environment at the same time also changes. At the moment that we're talking two monkeys doing foraging, now the sensory input, of course, for each of these two animals will change based on what the other is doing. And there's a lot of, prediction. Will that other animal come close to where I wanted to pick up my food? What is his intention? What is his preference? And so forth. So that is a level of complexity is unprecedented, and I don't think anybody in the world at the moment has a really good answer. But of course, what happened over the last 10 years or maybe a little bit more is that these recordings with high channel counts became much more common. And a lot of techniques have emerged to describe the dynamics that you can extract from those many channels as a dynamical system. And now we're getting back to physics, basically, and the mathematical tools that are relatively common in describing dynamical systems. And we've learned that sometimes the complexity of the underlying dynamics is surprisingly low-dimensional, as we call it. So it's actually not that complex. And even if individual neurons at some point contribute or don't contribute, that the overall dynamics are relatively stable. And they change in a meaningful way when animals switch between different types of behaviors, for example. And that is kind of the type of description that we're at the moment addressing. So what are the dynamics? What do they tell us about what the animal is currently doing or is up to doing? And that, I think, I'm not so concerned that we should not be able to easily see when an animal, for example, decides to take a turn to the left or to the right because there's higher reward here versus there. We have first evidence that this is actually extractable from the brain signals even while the animal is walking. And the brain is very busy with, producing the activity related to this walking. We still see on top of that what we consider the more cognitive signals. And since the neural substrate for both controlling movements and deciding which movement to make next is heavily overlapping, that will be still a major task to tell the individual components apart. And I don't say that we're there yet. But I think for most of the questions, there is already very good tools. But we're also collaborating with people who develop new tools, and I think that will be needed.
Yeah, the field is constantly, as I said, with this technology nowadays, and we can use maybe, I don't know, perhaps in your lab already some artificial intelligence to learn all of this. What are these activities? Can we see a pattern here? What is this pattern leading to? And etc. And it's extremely exciting to hear more about this in the future. Where is your stage of research? But I wanted to ask the following. So you mentioned the parietal cortex, where the sensory information is integrated, so to say, the motor premotor cortices, where we have the cells that tell us or move actually our limbs, and then the prefrontal cortex, where the planning and the higher cognitive functions are located. And in humans, we tend to plan in the long terms, right? We want to go to university, we think about it, and then our action is, so to say, uncoupled of our planning. And in animals, as you already mentioned, during foraging, chasing a prey, you have this instant decision making where you have to instantly decide I have to do this or that. In what to what extent is this planning of short planning different from the long-term planning, and can we maybe study now with all of this technology also the long planning in animals? So how can we actually say, okay, this was the long planning?
I think it's less a technological question. I think in terms of technology, of course, we could probably think of, well, we do have the long-term recordings. That is not really a limit at this point. We could also think of experimental paradigms where we encourage or require such type of long-term planning. I think for me, the more interesting question at that point is do rhesus monkeys at all have that type of long-term planning? And there's surprisingly little data on that. So in great apes or so, there has been more studies on that, and it's clear that there's a huge gap in terms of these types of cognitive functions between humans and non-humans. I do think we do observe still a gap. Of course, there's a lot of gradients in there, but we should be always aware that if we're studying a rhesus monkey, we are not studying a human. It's not the same. Mm-hmm. And the concepts behind these, what are needed for doing this type of long-term planning, have a lot to do with the concept of agency and how far do I see myself as the origin and the cause for things happening. And we see it in human development that over years that changes quite a bit. And then in the early years, the concept of agency that we're having is far from that adults have. Mm-hmm. And the same we see across species. There's huge differences, and certain animals just don't reach only a very rudimentary, like lower level of agency that we as humans, we can even, I can make an assumption that you are seeing yourself also as an agent and see myself as an agent, this recursive form of theory of mind, as it's called. We don't think that this exists in rhesus monkeys, for example. Mm-hmm. So in that sense, I'm not sure it would be the right model to study these long-term plannings and deciding, as a monkey, in which lab will I go. That's will be restricted more to the PhD students and postdocs, this type of decision. Now, and therefore, I'm always very aware of the limitations that we're having in terms of what type of cognitive behavior are we studying, and that's why these immediate so-called sensorimotor decisions, they are perfectly there. There's no doubt about it that primates have these skills, that they have goal-directed behavior, that they have, to a certain extent, planning behavior for the next few seconds or so. We can easily teach them this. And in that sense, they're the right animal model. I think that is up for discussion and for debate, in how far the mechanisms of arriving at a decision, the deliberation versus turning it into an action for these longer term decisions is actually the same or not. Intuitively, I would say, looking at other examples, how things evolved in evolution or evolve during development of a human, I don't think that the brain or the system developed something ad hoc completely new. There's no reason to believe that. So I do think it will build on this existing functionality that we see in sensorimotor decisions. And even the sensorimotor decisions that we're currently studying, actually in the recent years, are actually discussed more or less as a variant of motor control. Mm-hmm. Whenever I'm making a movement, I'm basically selecting one possibility out of an infinite number of possibilities. Mm-hmm. Any movement, any action that I'm doing is basically the result of a selection process. I'm rejecting other possibilities to make that movement. And in that sense, you could think of it as a decision process, as a selection process. Mm-hmm. And when I then attach certain values to action A versus action B, as it's typically done in value-based decision-making, these classical paradigms that come from economy, mm-hmm. right? Picking this product in the supermarket versus that product in the supermarket. This coming at a higher price is this, but maybe being more tasty. For this, I can reach conveniently straight. For this, I have to bend down. So all these costs and benefits that we're valuing against each other, in the end, they only show as overt behavior once you're making a movement. So in a sense, they are all precursors of selecting a certain type of movement. And then therefore, between motor control and these sensorimotor decisions, I see a continuum there. I don't see a categorical difference. And in the same way, I don't expect that we will see a categorical difference between short-term planning, let's say 5 seconds down the road, versus planning something for the next day or planning something for the next week. Mm-hmm, mm-hmm. In the end, until a decision of which university to go or whether to do my vacation in this or that country, it is always highly preliminary unless you take some form of action. And I don't think that we decide something in a definite way and then for 5 months do nothing about it. I don't experience this with myself. If I plan to go to a certain country for a vacation in 5 months down the road, of course it also comes with actions that I'm taking by the time I arrive at a decision. If it's only telling it to my kids, making a reservation, buying a travel book, right? We're translating these choices also into more closer actions. I don't think we ever do really decisions where nothing happens for months to go. I don't really think that this is the most typical case or scenario that our brains have built for.
Maybe the difference between us and monkeys is the sense of time, that at that time you will know I will go there. I don't know if they are capable of thinking about this. How can you study actually a problem like that?
This is an interesting question in terms of, in how far do we have a form of memory that points into the future. And I tend to conceptualize the signals that we're studying when I instruct an animal and then request the learned and associated action a few seconds later. I tend to refer to this as a movement plan. But in a sense, it also entails, on a more abstract level, a prospective memory, right? I need to not only memorize what my instruction was, but I need to memorize what my action goal is, what I want to do at some point. And in a more narrow sense, prospective memory refers to this possibility that we humans have, that we then also at some point remember that now we wanted to do it, right? I say I need to go grocery shopping. I need to get some milk and some bread. But it might be at a time when the stores are not open. And then I do something in between. I work on something. I completely forget about this. And all of a sudden, a few hours later, I remember I need to go get some milk and some bread. And this capacity of memorizing something that you want to do in the future and then recalling it actually and bringing it back into active working memory, that's a fascinating thing, because you're wondering, what happened to that information in the meantime? I think it's idling there all the time. It just doesn't, reach the surface of us making ourselves aware constantly, because it would basically, we have a very limited number of things that we can be actively aware of at any moment. So in terms of brain dynamics, I think it's just idling there and continuing as a dynamical process. And what then in the end triggers it to get back into the focus might be just the fact that, yes, from experience over many years, we learned if the pointers of the clock are in that range, it's more rewarding to go to the supermarket than going outside that time, because it would be open and not closed. So I'm sure there's additional sensory cues that then basically, based on experience, help us bring that dynamical process that stores that plan back to the surface.
It's very interesting. Also, when we're talking, I'm thinking now perhaps it's some circuitry that encodes this planning thing and keeps it somehow active, and you subconsciously, as you said, get input from some cues like the clock or someone reminds you or whatever, and then this kicks in and activates. Oh, I have to go to the store or book my vacation. It's very interesting in terms of how to approach this problem also.
I mean, we have seen in some of our recordings something that struck me as really surprising, and I was like, I didn't expect it, let's put it this way, and that was related to this topic. So before we had these large rooms in which the animals were free to move around for several meters, we had a smaller setup in which we also already let the monkeys move freely in a small volume, so they had to do 1 or 2 steps to be able to reach a certain target. So we instructed them which target to reach. Let's say I tell you, please use the right switch on the wall for turning on the light and not the left one. So we gave the animals this instruction, but they could not reach the wall yet. So they had to walk 1 or 2 steps and then touch the left or right switch on the wall. And when we observed the neurons in premotor cortex, there were neurons that at the time when I instructed the animal take the left or to the right, they would indicate that. They would be selective for left versus right. So it would fire at a high firing rate whenever you plan to go to the right target and at a low for left. And that selectivity was clearly visible. We had 4 targets. You could see 4 different levels of activity during that instruction phase and early planning phase. Then the animal had to walk there and then finally do the reach. And in some of these neurons, this information, whether left or right, this high or low firing rate, completely disappeared during walking. The neuron fell silent. And you think like, okay, if the neuron falls silent, the information is gone, right? You cannot distinguish it anymore between left and right if there is no difference in the activity. And by the time the animal got close to the target and did actually the reach, the separation showed again. So that, right, told me that, yes, the information, of course, is not lost. The animal knows what it wants to do. But individual neurons or parts of the network for a moment might not show it because now they're busy with some other task. Or they are getting silenced because the brain doesn't want those circuits to interfere with other ongoing tasks. But somewhere in the brain, of course, that information was stored. And by the time it became relevant, quickly before doing the reach, these neurons also started showing the selectivity again. So in the meantime, it must have been somewhere else. That's basically, on a short time scale of 2 seconds or so, something that I think is also possible for humans, at least, on much longer time scales.
Yeah, this is actually one of my main interests, how to understand this circuitry or the biology even behind this process, that how can neurons distinguish between things that we want to remember and what makes them forget, so to say, the information, and is it relayed somewhere else? What triggers this relay? And if you remember it and consolidate, you said it's a long-term memory, how do you trigger it again? It's these things we still don't understand, right? We have some sense about the circuitry, hippocampus, then the cortex, but the biology behind it, we have no idea what is happening, to my knowledge at least.
Largely, I would agree. I think it's a little bit more than no idea, but
a little bit.
At least I think what we have learned from very, very interesting work of colleagues of mine in the US, when looking prior to a action being really executed and implemented, when they we're wondering whether there is something like a gating mechanism, right? I'm having a plan already to do a certain type of action, but I'm, for some reason, not immediately starting with it. Sure? I'm blocking this type of activity. And one idea that is around for quite some while is that, okay, if at the cortical level we make those plans, maybe something more subcortical is gating that and just blocking it to have an effect on the spinal cord. Nobody really knew what the gating mechanism would be, but maybe there is something like that. And I think what we've learned over the last couple of years, you don't need such a gating mechanism. You can have, at the level of cerebral cortex, large-scale network activity being active the whole time, but in a way that it does not become active for downstream neurons. Those who read out from this brain network, while it's doing a lot of interesting stuff, while it's doing computation, while it's changing its activity, it just does not become relevant downstream. It's the so-called null space, it was called. Whereas in other phases, when the dynamical system switches to a somewhat different mode, all of a sudden, the changes in its dynamic do become relevant and do affect downstream circuitry. That's the so-called output potent space of the neural dynamics. And I find this a fascinating concept because it allows us basically to do a lot of what we traditionally call cognition, right? This magic thing that's between sensory processing and producing motor output, what brains are doing without becoming overtly visible to the outside immediately. This type of mechanism, if I may call it like this, allows you for doing a lot of computation and processing without immediately changing the configuration of your muscles and of your body and showing us actions. And then if you change the dynamics in a certain way, all of a sudden, downstream circuitry would basically pick up those signals and change its activity itself. And this is the type of thinking that currently, or since a few years now, is very prominent in motor planning / motor execution research.
So I have to update my information, definitely. Speaking of muscles and downstream, you're also involved in neuroprosthetic development in your research. Could you please elaborate on that a little bit more?
So, I mean, neuroprosthetics is both. It could be the thing where we record basically from the cerebral cortex, from nerve cells in the brain. Brain-machine interfaces are used to read out the patterns of activity from those cerebral cortex cells. And subjects, be it monkey subjects in the lab or being these days a few human patients in the US who are doing it already as humans, learn to control their environment based on the activity that they're producing with their cerebral cortex. Brain-machine interfaces. And we're doing this ourselves also. We have an animal who is used to work in a virtual reality, 3D virtual reality environment, and brain controls basically a cursor to move around 3D space. And the animal has learned a task similar to what I mentioned before, the center outreach task. It starts in the center and then moves the cursor to different positions, the corners of a cube. Now, this apparently is not a prosthetic device, right? This is just a cursor on the screen. Why are we doing this? We're doing it because one can use actually the logic of neuroprosthetics, this taking a set of neurons and their activity are now exclusively responsible for whatever action the animal produces. There is no other neuron that could have an impact on the action itself. And we use this as a tool for studying adaptation in neural circuits. We're using it as a highly controlled setting in which we can learn something about how the brain learns to control movements, where we have much more control over both who are the responsible neurons who are producing the movement and what is the type of feedback that the animal is getting. Because if the animal is not actually making physical movements, it's not changing anything of its somatosensory information. So the status of the muscles and all these signals that normally would come back when I change my body configuration, when I make a movement, this is basically held constant. And all feedback that the animal gets about the movement it produced is the visual feedback from controlling the cursor. And this we can control with a computer. Proprioception, the signals that come back from the muscles, we cannot control in an experiment very well. We can silence it maybe with drugs, but we cannot systematically shift it. Now, with this setting, we have a way of controlling exactly the feedback that the animals get and perturbing it in an experimental fashion. And we know who are the neurons who are producing this movement. And by this, we can basically apply perturbations and see how the motor networks change their dynamics to compensate for this perturbation. And that's the core mechanism in adapting our movements to changing environmental conditions and to changing sensory input. So we use it on the one hand at the cortical level as a tool for answering basic research questions. But as you mentioned, we're also working with clinical partners and partners from industry towards real prosthetic devices. I have to say in the recent years, we've paused those activities. These are activities that date back a few years. But back then our logic was, okay, highly invasive chips or microelectrode arrays, I prefer to call them, implanted below the skull in the brain is something where it's not really fully clear yet how well this will be accepted by patients, how eager they're to get those. If you listen to the people who received those implants in the US, they're super positively excited. At this point, I cannot say how representative this sample is, but in Europe, it's clearly from a political perspective, the medical field is much more reserved about this possibility of opening the skull, putting electrodes in, and using this as a control device towards controlling advanced prostheses. But what is very common already since many years is controlling limb prosthesis with muscle activity that is still available. So that, of course, would not work for a quadriplegic person, a person who is paralyzed in all 4 limbs. But let's say if you have an amputee who is missing the lower arm from some trauma, from some accident, and the rest of the body is still under the control of the patient, it still can send motor commands to other muscles, then you can pick up those motor commands from the muscles. And it's relatively comparably easy, at least, to record muscle electric activity. It's not as invasive as brain electric activity. You can record it from the surface of the skin. And that is something that is established since many, many years. It's in the clinic, you have a lot of patients who carry and wear those types of prostheses that are controlled by muscle electricity. The challenge that we back then were trying to address in communication with the companies who produce those products, they said that many of the patients are not so happy with it because the electrical resistance, the impedance between skin and electrode changes during the day when you're sweating, when the temperature changes. It's not the best signal to start with, and they were often frustrated about the impaired control that they have on the device. And you can imagine, if you're interacting in a social setting and the prosthesis is not exactly doing what you want, it feels uncomfortable. I took one of their prostheses once and were gluing myself electrodes to my skin. And then we went to one of these political events. It was an open house in the government in Berlin. And then comes the Minister of Lower Saxony and wants to shake hands. And I took this prosthesis and was shaking hand with the prosthetic device. And I was so scared that it would press too hard and that I have not enough control. Now, of course, a patient would have much more opportunity to control that than I had. But still, you want this thing to function. Otherwise, if you cannot rely on it, you put it to the side. And that's what many patients did. They put it to the side and decided and opted for a purely cosmetic prosthetic. Now, so that's why we set out to improve that. And the idea was, okay, it's not completely uninvasive anymore. But if we go below the skin, directly on the muscle, we're not suffering so much from changes in temperature, sweating, and things like that changes the impedance. So we get a much better signal-to-noise ratio. We can probably control much better because we can go on individual muscles, and the lower arm alone for controlling hand has many, many muscles, right? And what you record at the surface is a superposition of many of those signals. So the idea was, let's go on the surface of the muscle, and subepimysial, it's called, for the aficionados, and implant chronically electrodes there. But then, of course, we had the problem, now we need to get the signals out to the prosthesis. And having a wire going through the skin is A, aesthetically not so appealing, B, it's a hygienic risk that there might be an infectious pathway. So that's why we said, okay, let's go for wireless and transmit the signals through the skin. And that's what we then tested. That's where we came in. The partners from different universities and from the company were responsible for building the electronics for the wireless transmission. And we were basically training animals, rhesus monkeys, to get those implants and do their goal-directed reaches. That's what we are experts in, to train this to them. That was not a big deal. And then test in how far we get good enough signals from below the skin to the prosthetic device. Now, back then, it also was not really a prosthetic device, but we do some offline decoding of the signal. And it turned out it's just much more stable over time than whatever you can record from the surface of the skin. So in that sense, it was very promising. And then the project went into a phase where I said, okay, if we're now talking about longevity of electrodes, optimizing, the implantable devices, that doesn't really need a rhesus macaque model. We can test this in other animal models, which are not as valuable as a primate, and that's why we decided together that for a while now it will be tested in other animals before we might or might not go back to the primate model. I do think the next stage is probably directly go to humans. I felt so promising that one should directly go to human.
This was very exciting to hear about all of the research topics and all of the projects going on. And when you say so, I feel a little bit sad that I did not do rotation in your lab at that time when I had the opportunity, but the timing for me was not the great one. And however, I really want to continue talking about the science, but as I know, you have your tasks during the day and we have to finish at some point. So I would like to change the focus, and maybe since we are the German Primate Center, it would be meaningful to talk about animal research in particular monkeys. And you already said many times, several times, why it's important to address these questions in monkeys. But what do you do at this scientific center to address the well-being of the animals and to diminish, so to say, their suffering?
Yeah, so that's a very important question, and this is also something that from day one when I entered that field many years ago, it was for me clear that I personally need to be comfortable with the way we're doing the research. Otherwise, I would not do it. And that requires a very high level of responsibility. That's what I expect from all the members of my team. That's what I expect from myself. And as you said, or as I try to explain with my examples, I don't think that invasive research with animals anytime soon we can give up on if we want to understand the brain. I think it will be around for quite some while. And also these highly developed animals like macaque monkeys, rhesus macaques, or marmoset monkeys will remain important models, maybe even more important than they used to be now that we realize more and more the differences between rodents, and that translation to humans is often difficult unless you have also the primate model in between. So having said all that, I think we have one big advantage here at the German Primate Center, and that is that we're not only keeping these animals in small numbers for the immediate research need, but we are also a breeding facility, so we have very many of those animals. And we're having them since 40 years. So in that sense, what we have managed to achieve here is, and when I say we, I mean, of course, the people who have been there all the time and in other departments also, not just in our group, is a huge level of expertise. And that is just starting from the caretakers, via the vets, via the technicians, and the scientists. The scientists are the ones that are fluctuating actually at the highest frequency, right? The PhD students come and go, postdocs come and go. But what we try to create maximal stability, and I think that's one important measure, is with all the rest of the personnel. So the animal trainers, we have hired trainers who help with all these initial steps of getting animals integrated into our groups. We do group housing, right? Our animals live together with 2, 3, 4 other animals. The enclosures are much larger than that the EU legislation would require us to have. They have a heated indoor compartment, which is already bigger than needed. But then they also have the opportunity to go something that is semi-outside, so it's still protected from weather, but it has outside temperatures. And through all levels of interacting with the animals, we make sure that the people who are interacting with them don't change too often, so that they get used. They can build some level of trust, so to speak. They know what to expect. So you need to do this all in very small steps. That's also important. Don't confront an animal from one day to the next with a complete new situation. It will cause stress. There's no doubt about it. So we use what's called positive reinforcement training, so basically reward-based learning, to introduce them to new tasks. And at no point we will use any physical force or anything like that. So the animal, by means of understanding that if I do this, I get a certain type of reward. If I do that, I get a different type of reward. They learn to come from the large enclosure into a smaller enclosure. That smaller enclosure, we can push a little plexiglass in front of it. We call it the primate chair. The animals learn to climb into that chair and receive their reward. The animals learn that they stick out their head and let them be touched at the head, or later it will be then the implants. That's why we do this training early on, by means of being rewarded for it. And so you go step by step, always with familiar settings, with familiar people, so that they don't get too nervous about these steps. That's extremely important. And then once the animals are in the experiment, of course, you have multiple levels of assessing the current state of the animal. And that starts with the caretakers who see them every day. It's the vets that basically see them almost every day. It's the scientists who learn in bi-weekly discussions where we go through each and every animal and discuss the status of this animal, what to pay attention to. We do a lot of measurements on their weight development, on their consumption of fluids, on their fur condition, on their blood values. So it's a database with, by now, I think, millions of entries. We can, with one click, visualize their weight development over the last 30 days or the last 5 years, and you can see if something suspicious happening there. So it is this approach. Collect a lot of data, get to know your animal as well as you can, have people, the same people working with those animals over many years so that you notice when the behavior change. We have behavioral biologists who do also behavioral studies with them. And that's something that I wanted to add as a last point. We think we need to urgently move away when it comes to animal welfare questions from a highly anthropomorphic view that is quite common, right? People look at animals and think they have an understanding of what the animal experiences. And I think that's a very risky thing. Yes, with your own pet that you've been living together with for many years, I think you have a good feeling of whether something has changed and whether it's not feeling well. We try to get to this point with our monkeys as well. But of course, you also have sometimes new animals in the facility and you don't know them that well yet. So we still also need to be able to assess their welfare as well as with the others. And there, I think anthropomorphic view doesn't help us too much, and we need to approach this problem of assessing animal welfare with the same scientific rigor that we address all other research questions. And that's why our scientific director, Stefan Treu, and we are part of a consortium among other researchers in Germany who have really research projects on assessing animal welfare. And one of the things that we did there, for example, was that we took hormone samples, saliva samples, where we measured stress hormones in the context of an animal being in the experiment or not being in the experiment, and compared those. Turned out the fluctuations introduced by being in the experiment or not were completely negligible to the change in those hormones that you see over the course of the day between morning hours and late hours. So this was really minimal effects, basically no effects of the treatment. It told us, okay, apparently these animals were already super well accustomed to the situation, and that's why they didn't bother much about sitting in a chair or not. Other measures we took is, for example, cognitive testing. There is these famous, even in lay terms, we say, do you see the glass half full or half empty? It's called cognitive bias, and there is tests where you can test the cognitive bias, and we develop versions of that for animals. Also, attentional bias is sometimes used as a stress indicator. So we've developed machines that we can hang to the enclosures of the animals, touchscreen-based systems, kiosk systems, and the animals can go there anytime and work with this, and we can run those cognitive bias tests, for example. And we hope that by this we can have a warning system in future that if something shifts for an animal that we can look closer at it. And the last step that we did is to overcome, hopefully at some point, this anthropomorphic bias. Is we said, okay, what matters in the end is not how we think is less burden for the animal, but what the animal thinks. So we're currently developed a subjective severity scale that measures basically how much you would have to pay in terms of reward to an animal to opt for this version of the task versus this version. And these versions could be, for example, can I stay seated in my home enclosure and do the task on the touchscreen there. We would call it now mobile working. Or do I go into the lab as I used to go, with the transport being involved, with sitting in a room where I'm not anywhere close to my peers, right? I cannot see them, I cannot smell them, I cannot hear them. I'm in a separate room. What would the animals pick if they had the choice? Turned out there was a much more demanding project to get meaningful data than we hoped for, as often in research, for the reasons that apparently the differences for these animals for going to the lab or working with these machines in the local environment seems to be not very big. At least there was a lot of other factors that dominated actually their choice much more than this distinction. But by now we've recorded a few animals, and we think we have a way of titrating what preference would an animal have for which type of experimental setting. But we're talking the complexity of typical research projects, right? A PhD student being involved for 2-3 years before you get really conclusive data. But I think we need this level of rigor to make decisions about what are actually really the better procedures and what are not the better procedures. Because if we just go by intuition, we might sometimes not have the animal in the focus, but our view, and that might not be the correct one.
This is really fascinating. Basically, you have samples or probes or measures from every aspect: hormones, blood, behavior, physical appearance. You said now even these cognitive tasks. I was not aware about that, but it makes total sense, and it should be like that. And it also says something about your experiment, right? If you have a healthy animal, you perhaps have input that is desired rather than when you have an animal that suffers to some extent.
I mean, for the type of tasks that we are training our animals, you need a calm and relaxed animals. These animals would not learn those tasks if they would be upset or stressed out. That's the simple first thing that you would immediately notice. A stressed animal is not paying attention to anything that's happening there on this computer screen, right? It's just too far from the animal's need at that moment that it would pay attention to that. So you need to have these relaxed settings where animals can focus on what they're doing. Otherwise, you will not teach them anything.
Yeah. This is really, really fascinating. So we still have a couple of minutes, and I would like to hear your opinion on some of the problems in science. So we discussed with our previous guests some topics. For example, why do we have postdoc crisis, people leaving academia going to industry, or the reproducibility in some experiments, the burdens when you write grants and publish even. Could you maybe pick one aspect that you think is the most problematic? Of course, this has to do a lot with your research field, but maybe describe it and perhaps suggest some solution to this problem.
Each of these topics would be worth a full evening, I would say. They are all important. Maybe I keep this one short and then say a little bit more about another one. I'm not sure I would speak of a postdoc crisis. At least in the way the German academic system evolved over the last 20 years. We clearly, starting from the type of school degree that now more and more school students do, we broadened the base for who is entering university and who is going to do a PhD massively. And I think it was always clear we're not broadening this because we think we need so many more people in academia alone. But this is today, outside academia, there's a lot of jobs that require good academic education and maybe also the experience that you gain during a PhD when you learn to work independently, to learn to deal with problems and frustration and overcome it. These skills that you learn there, they're also interesting in other jobs and important for them. So I think the broadening of that base was never intended to all of a sudden have so and so many more professors in universities and postdocs and PhDs staying in that academic system. I think the idea was always that a significant amount of those people will move on and do something outside academia. And there is a lot of interesting research that's happening outside academia, right? Just think of all these companies who've created the vaccines in the last few years for us. So I think one should not see that as a failure if someone moves on outside of the system. That's what we should expect. And we should expect it, I think, for actually probably more people than actually stay. The problem I see only if that happens too late. So you should not be in the system for 5, 6, 7, 8 years as a postdoc and only then realize maybe I don't have a perspective. That's what we are responsible for as a university, as research institutes, as PIs, to make transparent that not everybody who starts with a PhD will end up in academia. And we should identify the alternatives and discuss with people transparently and openly what there are alternatives, in which way we can help with this, and then allow them to come early enough to a decision so that it's not so painful and doesn't feel like a failure. I didn't think it has to feel like a failure at all if done properly. So in that sense, yes, postdoc crisis, but mostly for the case that one is repeatedly fed with hope that maybe it works out in the end, and then it doesn't. That's a difficult situation. I agree with this. Now, one of the other topics you raised was reproducibility, and I think because I'm teaching also, I'm mainly teaching actually not in biology, but in psychology, so in biopsychology and cognitive neuroscience. And I think that field learned quite a bit over the last 10 years or so in terms of building protection against reproducibility crisis or problem, because there was a lot of cases where people felt like, hey, this is all not reproducible. If pre-registration of things in the end will do the trick, I think that will have to be seen. I think what it definitely reminds us of is, and that's actually quite a traditional thing to have in research, and maybe it got lost a little bit over the years, I don't know, but we need to be much more rigorous about the exact description of what we're doing, right? The methods parts of our papers maybe need much more attention than we hoped for. And I do see a significant challenge in this, because the level of complexity of what we're doing, the many stages that we're going through in preparation, in data collection, in data analysis, in combining many tools. Look at modern molecular biology papers where there's like 10, 15 authors on it, because so many different methods go in there. It is a significant challenge to describe all the methodological steps into the level of detail that someone could really reproduce it. And before that is not granted, it's very hard to say if there is a reproducibility crisis, in the sense that, okay, maybe things were just seemingly done in the same way and a different outcome was produced, but maybe they were not actually done in the same way. So that we need to be sure that we have a way of assessing that at all. Of course, nobody, blame myself here, is so super excited of digging through all these methodological details when writing papers, right? Think alone of the journals that put those at the end, right? You say, I go directly to the results, give me the meat, right? That's what I care about. But we cannot glimpse over the details. I think it has a potential of hurting the overall progress, because if we all are aware of this, right? If there's a result out there that turns out to be wrong, but was very influential, it's not just one study that failed, but it's this whole consequence of it, other papers being rejected because they're not consistent with this finding that was published so prominently. So you're slowing progress massively if that happens, that a wrong result is published. And I don't blame the people who did it. Maybe they had the best intentions whatsoever and they did it according to their standards in the field, but still it can happen, of course. this is one challenge. Will we find a way of publishing the stuff that it's really, really clear and transparent what exactly was done? It should be like this, but I'm sure in many cases we would have to realize it's maybe not. And the other challenge I see, and that's I think maybe even a bigger one, particularly in animal research. For primates, when I write my application of whether what type of research I want to do, I have to indicate whether this is a replication of something that is known already. In principle, I may not do something that is known already. That is not going to be approved easily. You have to have really, really good reasons why you would repeat something that might be known already. In fact, we spent most of the energy on writing those proposals of explaining why this is not known yet and why we need this type of research to get it approved. So, and I understand, of course, the motivation behind it. Nobody of us wants to use more animals than absolutely necessary for research, right? That's clear. We all agree on that. We should make sure we use the minimum number of animals. But every now and then, maybe a replication of a certain study is justified, and it is also legally possible, but the hurdles, I feel at least, seem to be significantly higher. For the reasons I mentioned, and these are reasonable reasons, but most people don't attempt repetition of a study. Also, of course, if you just show the same thing that has been shown of it, you don't gain much. You don't gain reputation by that. That's the third factor in this game.
Then it's connected with your grants,
Yeah, applying grants of saying I've repeated something that others did already before me is, of course, a difficult thing.
So definitely there are a lot of problems. And what is interesting to me that we more or less ask the same questions, and people have different perspectives. And I'm keen on listening to more of that, and then maybe put it together and analyze what are these burning questions that we have to address as young scientists, perhaps. So I look at this kind of a questionnaire that we prepared, and there are so many questions that I wanted to ask, but unfortunately we have to wrap up, and this room is going to be used in 25 minutes, and we have to put everything together back in the drawers. But one last question. What would you advise PhDs who would like to start a career in system neuroscience, particularly in your field, in motor, sensoric, and neuroscience, for example?
I'm not surprised about the question. I think it's a very difficult one because the way I run my lab is that I really try to pay a lot of attention of what the people bring with themselves. So the chance that there is this one advice, the one fits it all, it's not typically what in practice is so relevant. So when I have the meetings with my students and we're trying to find out what really motivates them or where they want to get at, it starts already with the ones that say, I know I don't want to stay in academia, and the others that say, no, I really want to do an academic career. And the advice for them is, of course, completely different. But if you, let's say, go with the model that probably many people enter academia with this goal of staying in academia and becoming researchers at a university and have a permanent position there, yes, don't be shy of trying out different things to find the thing that excites you most, because you will need that energy and this excitement to carry you through the years, and there will be years of uncertainty. I don't see that this will be in any way resolved. I think partly it's inherent to how we do science, and I would say the way we do science overall in terms of outcome is very, very successful. We learned so much about nature in the last decades. It's amazing. So it's not that we're doing something completely wrong in terms of producing output, but of course we need to make sure that we're not exploiting people on the way. So I think partly it is inherent that you will have to live with uncertainty. You should not be the person who is hoping for a super predictable 9 to 5 job where the tasks and the demands are laid out in front of you and this will hold through for the next 4 years. This is typically not how science works. You need to update, you need to be flexible, you need to react to publications by others, by discoveries that you didn't expect. So this flexibility you should have. And yes, there will be disappointments because experiments not always work the way. Sometimes others are faster than you. So you need to be prepared for this form of frustration. And I think nobody can really completely keep it away from you. So pick the topic you really burn for, and then it will carry you through those phases of slow progress or even setbacks. I think that's the only real advice I can give.
Yeah, I think it's a very valuable advice, and I thank you for that. And I hope some people will find it even more valuable and follow it, and follow the career path of academic, right? Professor Gail, thank you very much for your time here and for the very nice talk. I enjoyed it a lot. And as I said, I wanted to go more into science, but unfortunately the time is pressing us. But this is maybe an opportunity to meet again, maybe next year, and talk more about your project specifically. Thank you very much.
You're welcome. Thanks so much.
Neuroscience and beyond. Know more. Get inspired.